Well, hello, good afternoon, good evening, wherever you're tuning in from. My name is Simon Steele. I'm the Deputy Director of the Carl Sagan Center here at the SETI Institute. And before I introduce our special guest, Philipp Schiffer from the Worm Lab, I would say that the SETI Institute has always been fascinated by extreme creatures, whether on this planet or others. And so when we heard about a worm being defrosted and alive after 46,000 years, we thought we have to find out more about it. So welcome, Philipp. You're from the Institute of Zoology at the University of Cologne. Thank you for joining us. And I think the first question is, tell us about a worm lab.
Yeah, thanks for inviting me, Simon. It's very, very exciting to be here. The Worm Lab, it's basically my team of very enthusiastic people, and we are working with nematodes. And most people don't know what a nematode is. It's just a very small, tiny little creature, worms that live in the soil everywhere. So if you go, I see a tree behind you. If you go there and take some soil there, and you could find hundreds of specimens. And some people will have heard of C. elegans, which is a model organism, and which is used in laboratories all around the world. But our lab is the Worm Lab, because we are not working with C. elegans, but with all the very weird and fascinating different nematodes that we can find all around the world. We have a huge collection of them.
So these worms are everywhere. How do you define? If you go to look at a worm, how would you define a nematode worm? How are they different from other worms? What are their characteristics?
Yeah, so that's a very good question, actually, because if you say worm, that's actually not a taxonomic description of anything, right? So it's just form, right? So nematodes are within the broader group of the molting animals. So the phylum nematode, or the group of nematodes, is most closely related with arthropods. But of course, they have this vermiform body. They are very simply structured organisms, right? So no legs, no appendages on the outside. They have a cuticle, of course, as a barrier, and then inside their water pressure is keeping them in this worm form, right? So they have a so-called hydroskeleton, and then they have no real body cavity, right? So we call this a pseudocoelom, which would be the scientific term for it. Yeah, so really simple organisms, and they go through these molting stages. That's why they are molting animals, right? So you have larvae, and they have 4 larval stages, and then get your adult worm. And I guess you would have to see a picture to really see how a nematode looks like, right? And
yeah. So let's bring up a quick video of because this is the star of the show. So this is a nematode. There's no scale on this. Roughly how long is this little friend?
So this little friend is a millimeter in size, same size roughly as the model organism C. elegans. Most nematodes that we are working with are between this a millimeter, half a millimeter to a millimeter. Some are a bit longer, and most of them are actually of this size. If you look outside, or like in soil nematodes. But then there are nematode parasites, and the most fascinating one is, I think, 7 meters long, which lives in the renal system of a whale, right? And then there are animal parasites, which some people might have seen, even in the poo of their cat or dog, and they can also be longer than centimeters. So it's getting a bit freaky.
These creatures are literally everywhere.
Yeah, they're everywhere. So, this is the most fascinating thing. You can find them all around in every ecosystem. And although they are kind of like morphologically, quote unquote, simple, right? They conquered all of Earth.
Yeah. So definitely, some of you already told us where you're tuning in from. Wonderful, from New Jersey, from England, and from Germany. Keep those coming. Montana, Fayetteville, Arkansas. Please do tell us where you're tuning in from, and welcome. And if you have any questions, put them into the comments. Okay, we're going to go back in time a little bit. We're going to go back a little bit. We're going back 46,000 years, and obviously these nematode worms were in existence then. Do you know roughly how far back in geological time nematodes go? Is there an estimate?
So we don't have fossils, or there is, because of everything is soft body, right? So there is kind of imprints which people interpret as nematode fossils from 450 million years ago. But by just looking at molecular evolution, DNA, I would estimate that they're as old as arthropods, so 540 or a bit older, like when the Cambrian explosion was, and basically all the animal phyla evolved, and that would also be a time when nematodes evolved. In the ocean, of course, where you don't see any fossils of something that is a millimeter in size and has only a soft body.
Yeah, yeah. Take us back to Siberia 46,000 years ago. And also, why were you and your colleagues and your team looking around in Siberia for frozen animals? I mean, tell us the story of this discovery.
Yeah. So this is the most fascinating. I always tell this to my undergrad students as well, right? So because what basically happened is that the colleagues from they're working at the university in Moscow, and they were doing research in the Far East, in Siberia, in this Kolyma region, on permafrost. And you have these outcrops, which I guess some viewers know is kind of like just where you basically can walk up to a rock formation or some soil, in this case frozen soil, and they took samples from different time frames, and they can date them with the radiocarbon method, to do climate research, essentially, right? So they just took little soil samples, took them back to Moscow. That was in about 2003. And then over the years, as research goes, they thought little amounts of this soil to, look for pollen inside, do this radiocarbon dating to see basic climate histories through plants. And then, I think in 2013, 2014, they thought another tiny amount of soil, and we're really surprised that suddenly one of these worms crawled out, and actually two of them crawled out, two different species. We are focusing on one of them here, which we have analyzed. And they were able to date this piece of soil because there was kind of an animal burrow which had some carbon fibers and stuff close to where they found the soil, and they could date this to about 46,000 years ago or old. And so the conclusion then is that basically this worm has been frozen for 46,000 years.
Yeah.
And then the question is, of course, how, I mean, we can talk about this cryptobiosis, this being frozen in a second, but the other trait that's very fascinating with this species is that it's asexual, right? And so most nematodes are male-female, as most animals are. Some are hermaphrodites, but this specific species can reproduce asexually, so they're only females, which meant that this single worm, after having the luck of being taught after 46,000 years, could start reproduce. And then they grew a little culture, and they got in contact with people in Dresden in Germany, whom that kind of were collaborating already, and sent some worms over, and they got in touch with us because we are the worm lab and we are working with worm genomics. And so we teamed up and started to analyze how these animals actually can survive for such a long time or go into this cryptobiotic state, and also analyze the genome of these species, and also try to put them on the tree to see where they belong among all the nematode species.
Yeah. So you didn't you did radiocarbon dating of the surrounding material. You can't date the worm itself. It's just in the environment that is sitting.
Yeah. I actually discussed it with some geologists a couple of years ago, because I thought it would be so cool to actually date the actual worm. But they looked at me like, yeah, no, it's just too small, right?
Yeah,
so even with their most sensitive applying methods, they can't. The worm has literally 1,000 cells, right? And it's really, really tiny. So there's no chance of doing that. Yeah.
Okay, but so that would be really, really cruel irony to wake up after 46,000 years and then get turned into a lab experiment. When the worm was alive, what were the conditions like? I mean, was this worm living in frozen conditions and then it just happened to get frozen, or was it a different climate and then it got hit with something it wasn't ready for?
No, first off, it was like, I mean, through the last, I don't know, like 100,000 years or something, we had repeated ice ages, some smaller, some longer, right? And so I guess these worms were living. What we know from these worms today, they are living in like temperate climate, that also like where it's hotter, and just these species, they have this ability that when climate gets colder, they don't die off. They can go into the state of cryptobiosis, right? So basically, when we take them to the lab, if I put my worms into the fridge, for example, they start crawling much slower. And when we would put them into the freezer, they wouldn't. Some would die, and these cryptobiotic ones, they might survive, but they can't live in cold temperatures like this, right?
Yeah.
So they are adapted to what we call it North America or European temperatures. I get these species, right? And then when it gets getting colder, they just have this ability to survive this. Huh?
Yeah.
And I guess through the cycles of the ice ages, that must have happened repeatedly to this species. I mean, not to this individual worm, because it has been frozen for 46,000 years, but then when you thaw it, it still dies after 60 days, right? So it doesn't live for such a long time. It's just able to go into this abiotic state and then kind of survive frozen.
Yeah. So this is a wonderful survival trait. And there's lots of creatures that get frozen and they're not waking up. So what is special about these types of worms and other creatures that can, we know a lot about tardigrades as their ability to survive. That's our institute mascot, but what this isn't, we're not defrosting humans except in horror movies. So what's special? Is it because they're so simple? Is it because they have a sort of genetic makeup?
It's a genetic makeup. It's not. It's I was very hesitant to say simple, right? So because like a fly is not really more simple than a human, right? And also these nematodes, they have maybe a reduced morphology, but they're not from the genetics, for example, not simple, right? C. elegans has more genes than humans, but these animals which can go into cryptobiosis. So you mentioned the tardigrades; they always have the best press because they look cute. But then also rotifers, some midges, and some groups of nematodes, by far not all of them. They have this fascinating ability, and what we know is that they are using some biochemical tricks to preserve their tissue. So they enrich kind of sugars when they go into this state, especially one sugar which is called trehalose. And then I would say it is thought that this sugar helps them to protect their cells and their DNA from degradation. And already here I'm very careful because our knowledge almost ends there, right? So we know all these different animals that can be cryptobiotic, also some plants, of course. We know about this sugar and some other molecules that they use, but really the underlying genetics, how this all works, we don't know. That's, for example, something my lab is now working on, right? So one like part of our mission is we want to understand, like how does this very tiny creature realize, okay, oh, it's getting a bit cold, or it's getting a bit dry. I now need to switch on this whole genetic machinery and go into this state. And then also, like, how do they realize that they need to wake up again, right? And that they can reproduce again.
Yeah. Is there and again, this is probably difficult to say. Is there a limit? If you hadn't defrosted this worm, how much, without a natural defrosting effect, how is there a limit? Is there a sort of degradation over time that means that after 100,000 years, it probably would not structurally be able to be reanimated, for want of a better word?
I don't think so. So I think the limit is physical parameters. So, for example, background radiation.
Mm-hmm.
Other thing, I mean, they like now it is in a piece if of soil which is frozen, right? If there's a magma intrusion or something, or lava coming out next to it, or whatever, then it will just die, right? But I think once it's contained in this frozen state and just lying there and nothing really has happened, then I wouldn't know why over time. I mean, 46,000 years is such a long time.
Mm-hmm.
And then I think it could also go on for 100 or 150,000 years because nothing changes.
Yeah.
Yeah.
Okay. These extreme conditions that creatures are surviving. Obviously, it's not animated in that particular state, but you could end up with a sort of a thawing. Then the descendants will then freeze again. So you've got an extended lifespan over these extreme conditions, which is very, very exciting. I mean, potential for millions of years, maybe in the right conditions. So we're going to turn our attention to another part of the world quickly, the Atacama Desert, because the SETI Institute does a lot of work in the Atacama and looking at Earth analog regions, especially to compare with say Mars. Because if you look at where creatures can survive in the extreme conditions of the Atacama, you then have an idea of where you might want to look when you put your rover down on Mars, or ultimately humans. But that's a way away. Tell us what you were looking for and finding in the Atacama in these basically a very, very extreme desert.
Yeah. So basically, I mean, it's several things again, right? So just to make the connection to the frozen worms. So these cryptobiotic animals, as you will know, with the tardigrades is the same. Many of them, they can't only survive the freezing, but drying out as well. And that's basically because freezing and drying out or desiccation is the same process. It's that water is being displaced, right? And in the Atacama Desert, we look for first we look for diversity of animals that are there. We just had a publication on this where we looked in this very extreme environment that you just so nicely described, almost like Mars in some places, or maybe like Mars, I don't know. And just looked how many nematodes can we find there, because it wasn't described. And the answer is you can find a whole lot of them across very much the whole range of the Atacama. So up the highest mountains, or not there, but up to 4,000 meters to the sea level, and from north to south, you always find some nematodes, and they form distinct little populations. And then, of course, the second part of our project is again to ask, how do they survive there? And since in the moment, we can't go to Siberia and work there, and we have this research project in the Atacama. We are looking at cryptobiosis there and see which of the nematodes that we find there might do cryptobiosis, or which might find a different strategy to survive. Like they could be transported in and out by wind, by insects where they hitch rides. So looking basically at this evolution and ecology of nematodes.
Yeah. These are creatures, they're very, very small, and so you need to get pretty close to do that analysis on your hands and knees, and I'm thinking of a lot of missions to other planets. We've got a spacecraft going out to Europa at the moment, which is an icy moon.
Oh, cool. Yeah.
But of course, you're not doing a lander, and so is there? I'm thinking sort of stromatolites now, which are cyanobacteria mats, which are large macro structures for microscopic organisms. Is there any way of discovering nematodes without getting down on the hands and knees with a shovel?
No, you need a shovel. So basically, I'm used to carrying tens of kilograms of sand back from the Atacama or somewhere else, and then having my students and I dig through this, and like we're sitting with a microscope in the desert sometimes. I mean, wow. It's like they are tiny, and you can't. The main problem is not they are tiny; you can't see them, right? So because your stromatolites, they build like structures that you can see, but the nematodes, even when you look at a soil particle, like a tiny sand granule, and there was a nematode, and you wouldn't see it because you need to take the nematode off and look under the microscope. So it's a bit tricky.
Tricky.
Yeah.
So we got a few questions coming in from the audience. So they're looking at one from Ron. If the worm just comes alive again when it's warmed up, why has this experiment not been done before now? Does no one try this idea more recently? So is this revolutionary, or
No,
is
it's
this
not.
just a record breaker?
It's not. It's not revolutionary, right? So I mean, you said that it's known in other organisms, and people have been working on cryptobiotic animals for quite a while. That's how we know about this sugar, for example. It's just that nobody before had found such an old piece of soil from where then a nematode crawled out. Yeah.
Yeah. Okay. Question from Mathematica. I presume not your real name, but that will be cool. What do we know about the ancient genomes? What about more modern parasitic worms? So how 46,000 years, how similar or different is this nematode to ones that are being born today?
So that's actually something totally cool we are looking into right now. So it's a bit more nerdy details, but we knew that in the same genus, which is called Panagolimus, so this group of nematodes, that all the other species that we had before from, for example, California, also from China, Senegal, wherever, the sexual species, they all had 8 chromosomes. And asexual species, so which same as our permafrost form, they had 12 chromosomes. So call this triploid. They have an extra set of chromosomes. When we sequenced the genome of this nematode from the permafrost, we found it has 15. And it's really it's a Panagolimus nematode, so we can analyze this via morphology and with genetic data, and we see it's clearly a Panagolimus, but its genome is really different. And then in this genome, we see that parts of the genomes are mixed up, and then that's happening in this permafrost worm, but also, for example, in the worms from the Atacama, which are under extreme conditions as well. And in the moment, we are trying to understand how this is linked to cryptobiosis. I can say something about the parasitic worms if you like. I mean,
sure. Let's hear about parasitic worms. That's always good.
so I mean,
they're just living. They're doing their thing.
Yeah.
So we know yeah.
Important to know that these nematodes we are talking about now, the permafrost worms, Atacama, and the ones you find in your garden, most of them are not parasitic, right? They eat bacteria, some eat plants. They are very nasty human parasites, which you find especially in warmer regions. And with climate change, there's definitely the danger that they come up further north on the globe. And they are studied by many people, but there could be done more to develop drugs. Let's put it this way, right? So, but there is a lot of research on these parasitic worms, of course.
Yeah. Ron Schneider asks, how did you revive this worm, or does it just crawl out when you defrost? Well, was this soil sample defrosted to detect worms, or was it just, was that just a lucky?
Yeah,
lucky
just
discovery?
lucky. They were looking really like at the content of the soil, like for example, pollen from plants, so that they could date it and see what was growing back then. And they were really surprised when suddenly a worm crawled out of it. Yeah, and I mean also very lucky that they actually saw it, right? So because you can easily say, if you don't see it, it's gone. Yeah.
Yes. Yeah. And you've got to look out for these things. I mean, discoveries, it may be an accidental discovery, but you need to know what you're looking for to make an accidental discovery.
Exactly. Yeah.
That's fabulous. So I just want to, as we're coming up towards the end of time here, and if you've got more questions, please do add them in. So the worm that survived and is studied lasted, you say, a lifetime of, I forget, you said 40 days? Roughly?
Yeah, 40 to 60 days. It depends a bit on the species. Yeah.
But it was capable of self-reproduction. So
yes.
So the original star of the show is no longer with us, but
it went away a long time ago, like 10 years ago or something.
Yeah.
Yeah.
But the descendants are still alive and well and
I
being
have
studied?
them like in my lab, which is 5 kilometers from my home here. We have thousands of them, right? And we're actively working on them. Yes. Yeah.
Yeah, yeah.
Very happily reproducing.
That's good. Um, going back to Mars, there's
Mhm.
a question from Ron about could this type of nematode be on Mars or the icy moons or planets? I mean, I think the interesting thing about Mars is that it had a very wet climate very early on, and then now for the last 3 billion years or so, it's been very, very dry. So it is an Atacama-like. It sounds as though these worms are. If we did do a Mars sample return, which is another episode of
Yeah.
something altogether, um, it sounds as though you are not going to find fossil remains of this type of creature or the Mars equivalent of this type of creature. Is there any residual structure or chemical that it does survive in lieu of a fossil?
No, I don't think so.
Yeah.
And I mean, underlying to this question, you would, I mean, you already said this in a way, right? So we can't assume that if there was evolution of life on Mars, that it went the same track as here, right? So
Mm-hmm.
like that would be a very, yeah, big chance event, I guess. Yes.
Yeah.
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 Yeah. Well, where do you want to go next? What will be, as you say, these worms are everywhere,
Mhm.
but you're interested in their survivability and the extremophile versions of them.
Yeah.
Where would you like to go off and explore next?
So I'm flying to Namibia tomorrow,
Okay.
and I have also been to Australia. We are looking at Namibia, at the Namib Desert in Namibia, because as the Atacama is a West Coast desert, but the Atacama is a very old desert, and it hasn't changed for the last at least 30 million years, and it's hyper arid. Whereas the Namib is a bit more dynamic, right? It's younger and you see a bit more water influx,
Hmm.
so more frequent rain and the rivers there flood every now and then. So we do an ecological comparison between these two.
Hmm.
And then personally, I also went into the Australian outback, which is also a very, very dry place, but where there are places where there's seawater every winter. And so for me, it's basically interesting to um, see differences and similarities between species that I find in these three extreme places,
Hmm.
and then link it to the availability of water. And this kind of then extends again to a bit of change into research into global change, because of course some places are getting much drier right now.
Hmm.
And
Hmm.
so we hope that we can be able to predict something about evolution, and then linking to our cryptobiosis research, also maybe be able to find ways to make soil organisms more resilient in the face of global change. But
Right.
that's like a few years down the line, right? It
Yeah.
won't happen within the next 2 years in my lab.
Yeah. So there is that, science as a pure research is fascinating, looking at the history of our planet, looking at how our planet changes, and possibly looking at conditions and where to look for life on other worlds. But there is a more practical, if you like, application for this work in terms of
Right.
agriculture, in terms of disease prevention and things like that.
Yeah,
Yeah.
exactly. I mean,
Yeah.
many people, like I've been talking, when you talk to people about cryptobiosis and that these animals can, suspend their metabolism and quote unquote be between life and death, people asking like, can we freeze humans or something, of course.
Hmm.
And my answer to this is just like, yeah, I guess not really from our research on cryptobiosis, and if so, then maybe in 200 years.
Hmm.
But what you just said is much more tangible and imminent, that by studying this adaptation to extreme environments, we can potentially find ways to protect soils, which is so important for agriculture and life, our life, in the near future.
Yeah. And that's a good point to end. Philipp, thank you so much for joining us. Thank you, everybody, for tuning in online. It's fascinating that we're still learning so much about life on Earth, let alone life elsewhere
Thanks.
in the
Yeah.
universe. It's brilliant. Um, to remind everyone, the SETI Institute is a non-profit research center. We do rely on private donations to keep our research going. So definitely visit our website to see what other stuff is going on at the SETI Institute. We'll also, on the website, we'll put links to Philipp's work and his lab, so you can check out how the worms are getting on, which is wonderful. Thank you so much, everybody, and we will see you again soon.
Thank you.